This video is cell cycle regulation of cyclin CDKs by CDKIs and cell cycle arrest. This is figure 18-11. And so we're focusing on another type of regulator. It's called CDKI, which stands for cyclin-dependent kinase inhibitors. There's a variety of these out there, and we're looking at one in particular. It's this green blob called P27. CDKIs will bind to and inactivate cyclin CDK complexes. P27 specifically will bind to the G1 cyclin CDK, and then that causes an event called G1 arrest. Basically what that means is the cell is stuck in the G1 phase. To talk a little bit about the activity of this inhibitor protein, because this is new for our course, so P27 basically kind of gives the cyclin CDK complex over here, when they're active, this group hug, and so that inactivates it. And so I put a little group hug here, and basically this isn't exactly the same as a group hug because P27 is hugging this complex, though they're not really hugging it back. And again, this inactivates the complex. Another way to think of what P27 is doing, and might help you remember a little bit of the name, it has to do with a video I found on YouTube. So here's a zookeeper that was walking away, and this panda goes and grabs onto the zookeeper's legs to slow her down. And so maybe P for panda, P for 27, I don't know, might help you remember that. And basically this P27 here, the panda, is a negative regulator of progression to the next event. With that visual, P27 might seem kind of clingy, like it's kind of holding on to the old parts and kind of not letting the progression take place. However, P27 is actually a tumor suppressor. And so this is a normal thing that the cell cycle gets slowed down a little bit. I mean, it could get slowed down a long time, like a long goodbye, like another hug or whatever, and make things a little bit awkward. But the idea is that this is the brakes on the cell cycle for a tumor suppressor, and this can slow down the cell cycle. And that's overall a good thing. This slide is to talk some more about cell cycle arrest. And this is figure 18-12 in your textbook. It looks a lot like figure 18-3. However, notice that there's different information here. It's much more specific than figure 18-3. So be sure you are looking at figure 18-12. Also, your book doesn't have like a great definition of cell cycle arrest. They just call it a pause of the cell cycle and occasionally use the word arrest. But here's a definition. So the cell cycle arrest is the transient delay of progress through the cell cycle. And for example, this would be to pause at a checkpoint. And it's exactly where all of these arrests can take place, is at a checkpoint. I like to think of the cell cycle arrest points as kind of like a holding pattern. So if you've been flying in from some far destination sometime, and then you end up circling around the airport, for example, the airport's down here, just waiting to land, you're in a holding pattern. And basically it's like you're almost there, but not quite. Everything's in place, but you still got to wait for the last event to occur. That's the same thing for cells. So they can be in a holding pattern. So let's talk through a couple of these cell cycle arrest points. So first one is to talk about G1 arrest, which is right here. And that's at this restriction point. And basically G1 arrest occurs if there are insufficient mitogens, also known as growth factors, at the R point. And so that's what's going on here. And one way that could take place is through CDK inhibitors like P27. Then when there are sufficient mitogens, then the cell cycle can progress and just kind of pick up where it left off to go from G1 to S. The G2 arrest, so that's at the end of G2, and this is when there are errors in DNA replication. And then specifically CDC25, that phosphatase, blocks the entry to mitosis by not dephosphorylating MCDK. This is going to buy the cell some time to repair DNA, and so it can kind of hang out again in that holding pattern to get things fixed. And then if they get fixed, then it can progress forward. If it doesn't get things fixed, then again, apoptosis could be a possibility. For any of these checkpoints is the basic idea. So the M phase arrest point, so that's up here, and this is if there are errors in chromosome attachment and or mitotic spindle formation, and this event would be caused by inhibiting the role of APC, the anaphase promoting complex. So remember that normally helps with progression to end the M phase, can kind of keep moving through it. And so if APC is degraded, then that means it's still going to hang out in this holding pattern. This means that because APC is not active, then the M cyclin would not be destroyed, and then again that's going to buy some time for the cell to fix some errors. So let's sum up some of the mechanisms of cell cycle arrest, one of which is the presence of cyclins and or the concentration of cyclin. Then also phosphorylation or dephosphorylation of CDK complexes. Destruction complexes like APC are possible regulation points, as are inhibitory complexes like CDKI. And basically, on the plus hand, although this sounds like, oh gosh, that's kind of a pain, like being arrested sounds like that's a bad thing. The idea for the cell is that it's actually essential that the cell take the time to make sure that all of these events are taking place properly, because if it goes through this process too fast then and escapes the normal regulations, then this could lead to malignant cancers. And so overall that would be, again, life-death situation potentially for the organism. You'll see that there's a lot of breaks here. So basically the big theme for the cell cycle arrest is to slow everything down and basically slow down the cell cycle, allow some time for a good product to be made.